Hello and welcome to my YouTube channel, AnatomyMentor. Today we're going to discuss the histology of the kidney, and I have divided this lecture into two sections. In the first half, our learning objectives are that we are going to discuss the general architecture of the kidneys along with the microscopic structure of the renal corpuscles, and we shall correlate that with the process of blood filtration. And also I'll be discussing the microscopic structure of the JG apparatus and its role in regulation of blood pressure. So this is the first half of these two lectures on the histology of the kidneys. Let's take an overview of the urinary system and the general structure of the kidneys. Well, regarding the location and the extent, you should know that the kidneys are large, reddish bean-shaped organs which are present on either side of the vertebral column in the retroperitoneal space. This means only the anterior aspect of the kidneys that is covered by peritoneum. And each kidney, it is extending vertically from the level of the 12th thoracic vertebra to the 3rd lumbar vertebra. And the right kidney is positioned slightly lower than the left. So here is the front view of the urinary tract. This here consists of these paired bean-shaped organs, the kidneys, and like I told you, the right kidney is positioned slightly lower in comparison to the left kidney. And here is the concave medial border of each kidney, and this is the convex outer or lateral border of the kidney. Emerging from the concave medial border, this here is the area which is known as the hilum, where the blood vessels enter and where you can see the ureter, it exits from each kidney and it then transports the urine to the urinary bladder. And then it will eventually be extruded through the urethra. So these are the components of the urinary system. Now, what are the functions of the kidney? We know that they conserve body fluid and electrolytes, and they remove metabolic waste products, and they also have some endocrine functions. Regarding the dimensions of each kidney, it is about 12 centimeters long, 6 centimeters wide, and 2.5 centimeters thick, with, like I told you, a concave medial border, that is the hilum, and a convex lateral border. The upper expanded part of the ureter is known as the renal pelvis. Now, if we look at the internal macrostructure of a kidney, then in a hemi-sectioned, unfixed, fresh kidney, the outer portion is the reddish cortex, which is receiving about 90 to 95% of the blood supply, whereas the inner lighter portion is the medulla, which receives 5 to 10% of the blood supply. Now let's talk about the renal medulla first. So this is the area which is pale. It has striated renal conical pyramids, and their bases are positioned peripherally and their apices are converging to the renal sinus. And at the renal sinus, the pyramids project into calyces, and this part is known as the papillae. Now, the renal cortex is the outer part which is beneath the capsule, and it is the part which is arching over the bases of the pyramids. And extending between the pyramids are the parts which are known as renal columns. The peripheral lighter colored prolongations from the bases of renal pyramids into the cortex are known as medullary rays. So now we're going to look at these parts that we've just discussed in this simple diagram, which is showing the macrostructure of a hemi-section kidney. Over here, like I told you, the outer part beneath the capsule, this here is the cortex of the kidney, the renal cortex. And then the inner portion, this here is the renal medulla. In the medulla, you can see these here are the renal pyramids. 1, 2, 3, 4, these are the renal pyramids. And these pyramids are converging into this portion over here, which is the renal papilla. And eventually they will converge into the calyces, and the minor calyces will then merge into the larger 2 or 3 major calyces, which will then form the renal pelvis, which is the upper expanded portion of this here ureter. Now coming back to the renal medulla, which consists of these pyramids, well, between these pyramids, the portion of the cortex that extends between the pyramids into renal medulla is known as the renal columns. And we should keep in mind that the renal lobe is the portion of the kidney which contains a single pyramid with the adjacent area of the renal cortex. So this section over here is considered to be a renal lobe. Also, if we look at the hilum, which is present in the concave medial border, this is containing also the blood vessels, which are the renal artery and the renal vein. This dotted line over here is showing us the corticomedullary junction. Now, this is the hemisection of the kidney, which has been stained with hematoxylin and eosine. And if we look at it at the lower magnification, we can see this here is the outer cortex, which is covered by the fibrous capsule. And internal to that is this area, which appears to stain differently. This is the medulla. And you can see it consists of these pyramids. And here is the hilum, which contains the section of the ureter. Now, we know that the structural and functional unit of the kidney is the nephron. So we're going to discuss the structure of this part of the kidney, which is about 1 to 3 million nephrons in each kidney. And the major parts of each nephron are a renal corpuscle, proximal tubule, Henle's loop, distal tubule, and a connecting tubule. So these are the subdivisions of each nephron. The connecting tubules from several nephrons will merge or join with one another to form collecting tubules, and they will then merge as the larger collecting ducts. So this is a simple diagram which is showing us the structure of the nephron, which is the structural and functional unit of the kidney. So if you follow the laser pointer over here, I'm going to trace out the different parts of the nephron. This over here is the renal corpuscle, and this renal corpuscle, it in turn consists of a tuft of capillaries known as the glomerulus, surrounded by a capsule which is known as the Bowman's capsule. We'll be talking about its structure in detail later on. Let's look at the other parts of the nephron. Emerging from one aspect of the renal corpuscle is the proximal convoluted tubule, which is the second part of the nephron. And then comes this here looped portion, which is known as the loop of Henle. And then comes the distal convoluted tubule, which is then you can see merging with this connecting tubule. So these are the parts of the nephron. The connecting tubules of several nephrons will then lead into collecting tubules and larger collecting ducts. There are two types of nephrons based on the location and structure. So within the kidney, the first type are the cortical nephrons. These are the ones which are located almost completely within the cortex. And then are the juxtamedullary nephrons. They lie close to the medulla and they have long loops of Henle. Now, this is the histomicrograph of the kidney at a lower magnification. So we can see several parts of the nephron. Over here, we can see the corpuscles. These are the most prominent ones, which we can see over here, having a central tuft of capillaries surrounded by the Bowman's space and the capsule. So collectively, this is known as the renal corpuscle. You can see several renal corpuscles in this histomicrograph, and other sections are through the collecting ducts, the different parts also through the tubules, which could be proximal or distal convoluted tubules. And if we generally look at the region, this here is the cortex, and over here, this here is the medulla. The collecting ducts are present over here, and this is, you can say, a panoramic view of the microstructure of the kidney. Now let's focus on an individual renal corpuscle. I told you it's a spherical structure. It has an average diameter of about 200 microns. Now it has two poles, a vascular pole and a urinary pole. The vascular pole is where the afferent arteriole enters and the efferent arteriole leaves, whereas the urinary pole is where the proximal convoluted tubule begins. Now, the renal corpuscle consists of a tuft of glomerular capillaries, which are surrounded by a double-layered Bowman's capsule. So there are two layers of Bowman's capsule. The inner one is the visceral layer, and the outer is the parietal epithelial layer. And between these two layers is the space which is known as the urinary or the Bowman's space. So here let's look at the structure of the renal corpuscle. This is the tuft of capillaries, and this is formed by the afferent arteriole, which is the one which is entering the renal corpuscle, and from the other aspect, you can see the efferent arteriole emerging from renal corpuscle. The afferent arteriole, you can see, is of larger diameter in comparison to the efferent arteriole. The part of this renal corpuscle where the arterioles are attached, that is known as the vascular pole. Now let's look at the other aspect, the other side of the renal corpuscle where the proximal convoluted tubule begins. This here is known as the urinary pole. And the tuft of capillaries, this is surrounded by the Bowman's capsule. Bowman's capsule has an external layer, which is the parietal epithelial layer, and it has an inner layer, which is the visceral layer. It is in close apposition. It is lying closely attached to the glomerular tuft of capillaries. And between these two layers, this here is the Bowman's space, which contains the filtrate. If we want to understand how the layers of Bowman's capsule are arranged or organized, this is a very simple series of diagrams which shows us the development or formation of the Bowman's layers. So you can see here is the tuft of capillaries, that there's a developing glomerulus with the afferent and the efferent arteriolar ends. And we can see that this here is the tubule, which is lining the tubular cells which are lining the primitive renal tubule as this glomerulus. It extends into the Bowman's capsule. The inner layer is going to be closely opposed to tuft of capillaries. This is forming the visceral layer of Bowman's capsule, and the outer layer will be present on the external aspect as the parietal layer of Bowman's capsule. So this is the visceral and this here is the parietal layer, and between these two layers, this here is the Bowman's space. Now, what is the filtration apparatus? Well, filtration apparatus is also known as the glomerular filtration barrier. So this consists of three components. Number one, the endothelium of glomerular capillaries. Number two, the glomerular basement membrane. And number three, the visceral layer of Bowman's capsule. So over here, we are looking at another diagram of renal corpuscle, this time in a little more detail, in which we can see this is the tuft of capillaries, and you can see they're surrounded by the visceral layer of the Bowman's capsule, and it is present in the form of these foot-like processes which are known as podocytes, and podocytes further have these secondary processes known as pedicels. And on the external aspect, this here is the parietal layer of Bowman's capsule. Between the parietal and visceral layers, this here is the Bowman's or capsular space, which will contain the filtrate. We know the blood is going to enter the tuft of capillaries from the afferent arteriole, which you can see is of larger diameter in comparison to the efferent arteriole, which is of smaller diameter, where the blood is going to exit or leave the glomerulus. Now, while the blood travels within this tuft of capillaries, which is known as the glomerulus, the blood is going to be filtered across the glomerular filtration barrier. And as I told you before, the glomerular filtration barrier, it consists of these three components, which are the endothelium of the glomerulus, and you can see the glomerular basement membrane, and then, of course, the visceral layer of the Bowman's capsule. And eventually this filtrate is then going to enter the proximal convoluted tubule at the urinary pole of the renal corpuscle. So these arrows in black are showing the direction of the blood flow, and these arrows in blue are showing the direction of flow of the filtrate. Cells of the visceral layer of Bowman's capsule, so they're specialized, and I told you they're known as the podocytes. They have primary processes which give rise to the secondary processes, which are pedicels. Now, the gaps between the adjacent pedicels or secondary processes are known as filtration slits. Each is about 25 to 30 nanometers wide and spanned by thin diaphragms. The endothelium of the glomerular capillaries is fenestrated, which means they have windows but without a diaphragm. The glomerular basement membrane is formed by fusion of the basal laminae of the endothelium and podocytes, that is the cells of the visceral layer of Bowman's capsule. And collectively, as I mentioned before, they're going to form the filtration barrier, or we could say simply the barrier across which the blood is going to be filtered. So this here is now a diagram which is zooming in on the filtration barrier. This here is a single podocyte. This is the cell body of the podocyte emerging from the podocyte. These here you can see over here are the processes of the podocyte, the primary processes. And then from the primary processes, these here are the secondary processes, smaller ones known as pedicels. Between them are the gaps which are known as the filtration slits. And across the filtration slit, you can also see this here is the glomerular basement membrane, and then you have the fenestrated capillary endothelium, forming the filtration barrier or filtration membrane. This again is the same diagram showing us the three components of the filtration barrier. You can see this here are the secondary processes or pedicels, the capillary basement membrane, and then the endothelial the fenestrated capillary endothelium. And this is showing this same filtration barrier in relation to the function, that this here is the blood within the capillaries. It is going to be filtered across these three components of the filtration barrier. And here the ultrafiltrate is then going to enter into the Bowman space. So here we can see the three components of the glomerular filtration membrane or barrier. First, you can see the fenestrated capillary endothelium. By fenestrations, we mean that there are gaps or spaces within the endothelial cells. Then the second component of the glomerular filtration membrane is this here, glomerular basement membrane. This is the second component of the filtration barrier. And then the third one is this here. These are the filtration slits which are present between the pedicels or the secondary processes of the podocytes. So these are the three components of glomerular filtration barrier. And this is the barrier across which the blood is going to be filtered. And we can see the arrows there are pointing towards the direction of the formation of the filtrate. This here you can see is the blood within the capillaries, and it contains the leukocytes, the large proteins, platelets, erythrocytes, which of course will not be able to cross the glomerular filtration barrier, but it does allow the smaller proteins to pass across this barrier, and a few of those small proteins will be blocked along the way, but eventually the filtrate will contain, you can see, water, glucose, amino acids, ions, urea, many hormones, vitamins, B, C, ketones, and like I mentioned, very small amounts of protein. So this glomerular filtration membrane or barrier is having the function of a physical barrier to the larger proteins, and also it's an electrochemical barrier because the anions are unable to pass because of the negative charge of polyanionic glycosaminoglycans in the glomerular basement membrane. So the GFR, that is the glomerular filtration rate, remember, is constantly regulated by neural and hormonal factors that affect the degree of constriction in afferent and efferent arterioles. The average glomerular filtration rate is 125 ml per minute or 180 liters per day. Therefore, the entire blood volume is filtered about 60 times every day. Now, you know that in certain diseases such as diabetes mellitus and glomerulonephritis, our glomerular filtration barrier becomes disturbed, and so its permeability is altered, and many proteins are able to pass through this barrier, and then they will appear in the urine, and this condition is known as proteinuria. Proteinuria is an indicator of many kidney disorders. Now let's talk about the role of another cell population in the kidney, which are the mesangial cells. Mesangial cells, they provide physical support to the capillary endothelial cells, and they also perform the function of phagocytosis of protein aggregates, and they have a secretory function. They are producing cytokines, prostaglandins, and extracellular matrix components. And the long contractile processes of mesangial cells are closely associated with the blood capillaries within the kidney, and therefore they maintain optimal filtration rate at the glomerular filtration barrier. And they respond to the changes in blood pressure. Here we can see a diagram which is showing us the endothelial cells surrounded by the podocytes, the primary and secondary processes. And over here also we can see the mesangial cells are present in the mesangial matrix. This over here blue represents the glomerular basement membrane. Now this is the histomicrograph of the renal corpuscle at a higher magnification. So we're looking specifically at the renal corpuscle over here, and we've used a special stain, which is periodic acid shift stain, which is going to stain the basement membrane magenta pink color. So this here over here you can see is the outer parietal epithelial cell layer of the Bowman's capsule, and these are the nuclei of the epithelial cells in the parietal layer. And this here is the Bowman's space or the urinary space. Then this is the tuft of capillaries, that is the renal glomerulus. And these are the endothelial cells. Over here you can see these are the endothelial cells, the nuclei of the endothelial cells. And this here is the glomerular basement membrane. And you can also see the capillaries. You can see the mesangial cells and their nuclei over here within this glomerulus. Okay, so here is the afferent arteriole, and I told you before the arteriole is present at the vascular pole of this renal corpuscle, and this here is the section through the proximal convoluted tubule. You can see the epithelial cells in its wall. So this is the urinary pole of the renal corpuscle. Now we're going to discuss the microstructure of a very important part of the kidney, which is the juxtaglomerular apparatus. So the JG apparatus is a specialized sensory structure that utilizes feedback mechanisms to regulate glomerular blood flow and keep the glomerular filtration rate relatively constant. This is the function of the JG apparatus, to regulate the blood flow through the glomerulus and keep the glomerular filtration rate at a constant level. Adjacent to renal corpuscle is the location of the JG apparatus, and it consists of three components. The first component are the specialized smooth muscle cells of the afferent arteriole, which are known as the JG cells. Second component are the specialized cells of the distal convoluted tubule, known as macula densa. And the third component are the extramysangial, the extraglomerular mesangial cells, or the lacis cells. So let's just see the location of the JG apparatus. If you remember, I showed you this diagram before. This is the nephron, and these are the different parts of the nephron. This here is the location of the JG apparatus, which consists of a part of the distal convoluted tubule and the afferent arteriole. And if we look at then again the diagram of the renal corpuscle, we will see that this here is the vascular pole. Here is the afferent arteriole, and these are its specialized cells known as the JG cells. And adjacent to it is a portion of the distal convoluted tubule. And here in its wall are the specialized cells known as macula densa, the second component of JG apparatus. And this here are the extraglomerular mesangial cells, or the lacis cells, which are the third component of JG apparatus. So 1, 2, 3. These are the three components of the JG apparatus, which you can see over here are visible, and their location is just adjacent to the renal corpuscle. Then again, the diagram of the renal corpuscle, which is showing the JG apparatus adjacent to it, consisting of this here, you can see the JG cells and the lacis cells and the macula densa cells. And if we look at the histomicrograph at a higher magnification of the glomerulus, well, this here is the glomerulus with the central tuft of capillaries surrounding Bowman's space and parietal layer of Bowman's capsule. And this here is the vascular pole. And over here is the afferent arteriole, its specialized JG cells, and also are the lacis cells. And this is the macula densa of distal convoluted tubule. These are the three components of the JG apparatus, which you can see in the stain section of the renal tissue. Now, how does the JG apparatus work? Let's talk a little about the function of the juxtaglomerular apparatus, and we can discuss this briefly with the help of this flow chart. You can see over here that these are the three components of the JG apparatus. Now, what happens in response to a decreased blood pressure? So if the blood pressure is decreased, obviously it's going to cause a decreased glomerular filtration rate, which in turn will cause a lowered sodium concentration in the fluid of distal convoluted tubule. Now, sensitive to this, this lowered concentration of sodium, we have the osmoreceptors, which are the macula densa, they're osmoreceptors. and they're now going to cause release of a substance known as renin from the JG cells. Also keep in mind the JG cells, which are specialized cells of afferent arteriole, are themselves mechanoreceptors, or we can call them baroreceptors, which will also be responding to the decreased blood pressure, causing the release of renin. Now, what is renin? Renin is a protease. It means it's going to cleave a protein which is known as angiotensinogen. It is a plasma protein. It's going to cleave it into the decapeptide, which is the angiotensin 1. Angiotensin 1 is then going to be converted into the active form angiotensin 2 by ACE, angiotensin converting enzyme found in the lung capillaries. Once we have angiotensin 2 is itself a potent vasoconstrictor of blood vessels. So as it constricts the blood vessels, it causes the blood pressure to be increased. And it also has two other functions. It causes the release of aldosterone from the adrenal cortex, which increases sodium uptake from the distal convoluted tubule, which in turn increases water uptake, and of course that will lead to an increased blood pressure. Remember, angiotensin 2 also has a direct action on the renal tubular cells to cause the increased sodium uptake. So this is the basic working of the JG apparatus, and in maintaining the glomerular filtration rate and in maintaining the blood pressure. So you can see there are different components of this apparatus working at the functional level. Now that I've discussed the basics of today's topic, I'm going to just discuss a few MCQs with you and a little histology quiz at the end. So the cell type that comprises the visceral layer of Bowman's capsule, if you recall, that is the podocytes. E is the answer for this one. And then the modified smooth muscle cell that secretes renin so renin is produced by, keep in mind, the JG cells, the juxtaglomerular cells. Okay, so let's take a brief quiz of histology. After discussing the microstructure of the kidneys today, you should be able to identify these parts of the lobe of the kidney. So this is a renal lobe, and over here you can see number 1 is the renal corpuscle, which is the part of the nephron, and within that renal corpuscle, number 2 is the glomerulus, number 3 is the proximal convoluted tubule, and you can see number 5 here is the loop of Henle, 4 is the distal convoluted tubule. So these are all the parts of the nephron, which is a structural functional unit of the kidney. And then number 6 over here is the collecting duct of Bellini. And this portion of the renal pyramid you can see over here is the renal papilla, number 7. Then another diagram over here is showing us the details of the renal corpuscle. Number 1 is the proximal convoluted tubule, which is entering at the urinary pole. Number 2 over here are the podocytes, which is the visceral layer of the glomerular capsule. And number 3 is the parietal layer of glomerular capsule. 4 is urinary space. 5 is the afferent arteriole. 7 is efferent arteriole. And 6 over here is the JG apparatus, which we talked about in detail. The different components are, you can see the macula densa, the laces cells, and the JG cells. And here is a micrograph showing us the JG apparatus and the renal corpuscle. Now let's look at a few electron micrographs, and in that particularly the glomerular filtration barrier can be visualized in detail with the help of an electron micrograph. So number 1 over here is the podocyte, number 2 the urinary space, number 3 you can see are the pedicels, that is the secondary processes of the podocyte, and then number 4 you can see is the blood within the capillaries, and number 5 over here, if we look at it in a higher magnification, these are the filtration slits which are present between the pedicels. Number 6 is the glomerular basement membrane, and you can see number 7 over here, this is the fenestrated capillary endothelial cell. So these are the 3 components of glomerular filtration barrier. I'm going to repeat them. You can see the filtration slits between pedicels, that is number 1. Number 2 is the glomerular basement membrane, and number 3 over here is the fenestrated endothelial cell. So these are the 3 components of the glomerular filtration barrier which are visible on the electron micrograph. So this concludes with today's discussion on the histology of the kidney, in which we discuss the microstructure of the renal corpuscle and also the microstructure of the JG apparatus. And if you like today's lecture, if you did like today's discussion, you found it helpful, don't forget to like and subscribe to my YouTube channel, AnatomyMentor. You can simply do that by pressing on my picture, which is going to appear at the end of this video presentation. So I'll be coming soon with the second part of this discussion on histology of the kidney, in which we'll be talking about the convoluted tubules, the histology of the convoluted tubules in a little more detail. So stay blessed and stay sharp. Take care.